today. We ended last week with chemistry. We'll finish up the last little bit of that. And that's going to lead us into more chemistry as we start to think about DNA structure. I wanted to, and then once we get through that, we'll start with mitosis. Before we get into DNA structure, I just want to do a little bit of homework review. I talked to TIs last night, and I want to make sure there seemed to be some confusion on some of the questions for homework 1. I want to make sure that we're all clear with some of that information. So I'm just going to do a few quick review questions. So you had one question in homework 1 about water. The question was, a single molecule can form non-covalent hydrogen bonds with how many other water molecules? 4. Why 4?
The oxygen bonds with 2 other hydrogens, and each hydrogen bonds with an oxygen.
All right. I had a clicker question, but you guys answered it already, so I won't make you do that. All right, so there seemed to be a little bit of confusion, so I just want to make sure everyone is clear. All right, hydrogen bonding in water. One hydrogen can bond with 4 other waters based on the distribution of those charges. Okay? So just make sure everyone's clear on that. This one I will ask you as a clicker question. Let me open up the poll. So I've opened up a poll. I want to make sure everyone is clear about what peptide bond is. We'll cover it a little bit later in the semester. But I want to make sure there's no confusion between a peptide bond and some of the other bond types that we're covering right now. Everyone get a chance to answer? Take a look at what the class thinks. Oh, the results are hidden now too. Add this to my list of things to do at the beginning of class. All right, so still some confusion. Right, what is a peptide bond? Defend your answer. You just guessing randomly? Anyone want to justify what they chose? What is a peptide? All right, what's an acid plus acid?
Anhydride.
What's an acid plus an alcohol? What are those other 3 choices? All right, so we know it's not A or B. What's a carboxyl group plus an acid? What's a carboxyl group? Another way to call it? It's an acid.
Carboxylic acid.
So an acid plus an acid is a anhydride, so it's not E. That leaves us C or D. We're down to 50-50.
Let's see.
Why C? What is an example of a peptide bond and what biological structure? Protein between amino acids. All right, so the correct answer is C. It's a carboxyl group and an amine group. So the end of one amino acid and the front of another is an example of a peptide bond. All right, so keep that in mind. We'll get to proteins a bit later in the semester. All right, so here is where we left off on Friday. All right, so just a little bit of review. I wanted to make sure everyone was clear on for the homework. All right, so we left off here in ester bond, condensation reaction between an acid and an alcohol. We looked at an example of that in a biological molecule, this nucleoside monophosphate. All right, and in this particular case, it's a phosphoester bond, right, because that central atom is a phosphorus, not a carbon. This is CMP, cytidine monophosphate. All right, we'll get to the naming conventions in just a minute. We had one more bond type to cover. This is a give me, all right, give you some points. If you were paying attention last Friday. Which one is the anhydride bond? Get a chance to answer? Go ahead and see what everyone thinks. All right, E. Excellent. I think we're clear on anhydride bonds for the most part. Let's just review really quickly. So what kind of bond is A? What functional group is there? It's an amine. What kind of bond is B?
And glycosidic.
Glycosidic. What kind of bond is C?
Carbon-carbon.
Carbon-carbon. I know you were looking for a more complicated answer, weren't you? What about D?
Ester.
Phosphoester. And what about E?
Phosphoanhydride.
Phosphoanhydride. All right, so you should recognize each of those bond types in the exam. All right, so anhydride bond, how would we define it? We already did that this morning. I heard it sort of whispered. Acid and an acid with that characteristic structure. All right, so if we bring our another phosphate group into our molecule, right, notice they've given up those hydrogens, therefore acid. Bring that in, have a condensation reaction, anhydride bond. Again, because we're using phosphorus instead of carbon, the phosphoanhydride bond. All right, so this now is a CDP, cytosine diphosphate. Two phosphates on there. And just to show you that on anhydride bond structure relative to what we have put together in our nucleotide. All right, so you must recognize the difference between a phosphoester and a phosphoanhydride bond in biological molecules. Hopefully at this point, you guys have had lots of practice with that. We can add another phosphate group on there. We're now at cytidine triphosphate, CTP. How do we refer to each one of those phosphates? All right, red, orange, and yellow. Alpha, beta, gamma. All right, so we now have the building block of DNA, nucleotide triphosphate. that leads us, now that we've put together our building block, let's put those building blocks together into nucleic acids. So for naming, you need to know what a nucleoside is, what is a nucleotide, and how do they differ. What's a nucleoside?
Bases plus the sugar.
Just the base plus the sugar. What's a nucleotide?
Phosphate group, base, and sugar.
Nucleoside plus a phosphate. All right. So let's do a few quick, just shout out the answer kind of questions. So we already answered this. Is the nucleoside. Why? No phosphates. Right? It's just the base and sugar. What's the name? Adenosine. Good job. All right, that looks a little bit familiar from your homework. All right, so the base plus the sugar, adenosine. What's the name of just the base?
Adenine.
Adenine. Is it a purine or a pyrimidine? Purine. Why?
Has
Two
it
rings. Purines have 2 rings. So what are the pyrimidines?
They're guanine and cytosine
Cytosine,
and thymine.
thymine, uracil. Thank you. I almost forgot about that third one. All right. So this is all should be review. So what nucleoside is this? Trick question. Look again.
Thymine. Deoxythymidine. I don't think
Deoxythymidine.
that's how you say
Right? What hydroxyl group is it missing?
it. 2 things.
The 2 prime. Which one is this? Uracil is uridine.
Oh.
Right? Uracil is the base. I've got one more. Which one have we missed? Guanosine. Thank you. All right, so you need to, as you probably guessed from your homework, know all of these backwards and forwards. You need to be able to draw them. You need to pay attention to whether we're talking about a base or a deoxy base, a nucleoside or nucleotide. All right, so if I say draw cytosine, you would draw, cytidine, you would draw cytosine plus the sugar, no phosphates, right? So the naming convention, nucleoside equals a base plus a sugar only. It becomes a nucleotide when you start adding phosphates. The nucleoside triphosphate, as I've already mentioned, is that building block for making our DNA. So you need to know all of these. All right? What they're called. So if I ask for guanosine versus deoxyguanosine, or if I ask for any of these with a mono, di, or triphosphate, should be able to draw them. All right, so start practicing now. All right, so those are all our bases and nucleosides. We add some phosphates to become our nucleotides. So let's start putting together these single nucleotides into our nucleic acid strands. What kind of bonds connect single nucleotides into a nucleic acid polymer? All right, think about Friday and what we covered earlier today. Talk to each other. All right, so we're just building a single strand and we're putting nucleotides together. Right, single strand. All right, how do those nucleotides fit together? What two things are reacting to form the bond between nucleic acid or nucleotides? Right? You're going to link two nucleotides together into a strand of DNA or RNA. What type of bond is that linking those two individual nucleotides?
Yeah, but they can like keep it together or this in a or time or it's going like this.
All right, I'll give you one more second. Get your answers in. Single strand. Yep, interaction between the two nucleotides to form the single strand of a nucleic acid. Everyone had a chance to answer? Take a look. Yes. If we're talking about, from now on, we'll probably be talking about DNA or RNA. So, yes. Unless I specify differently. So the question is whether or not if I say deoxyribose, you should assume it's 2'. So in your homework, I had you do a few different varieties of deoxy sugars. Now we're talking about DNA and RNA. So in that context, right, DNA is always 2' deoxy. Okay? All right, so. Got off the screen, or is that? Okay, there we go. All right, so the majority of you got that correct. It is an ester bond that links the two nucleotides in a single strand of a nucleic acid. All right, let's take a look at that a little more closely. So it's actually a phosphodiester bond. Right, so there's an ester bond on either side of that phosphorus molecule. What two carbons are those phosphodiester bonds between in one nucleotide versus the other? Right in the front, anyone in the back want to yell it out? 5' carbon and 3' carbon. All right, the linkage always happens in the same place. All right, so take 2 minutes and draw me nucleotides with that phosphodiester bond. All right, practice, practice, practice. Get as much of it as you can.